Yeast is one of the oldest laboratory model organisms known to man, and the amount we've learned about human biology from this single-celled creature is simply amazing. While there are many types of yeast used in research, one of the most popular is Saccharomyces cerevisiae. This type of yeast is mostly known to humans because it's used for making bread, wine, and beer. It's not harmful to humans, and it's extremely easy to grow. All it needs is some kind of sugar and sources for nitrogen and phosphorus. It can grow either in liquid cultures or on solid surfaces. When grown at 30 degrees Celsius, the number of yeast cells will double every 1 or 2 hours, but keeping the cells at 4 degrees Celsius will almost completely prevent their growth. All living organisms are made of one or more cells. Animals and plants are all multicellular organisms, while bacteria, amoebas, and yeast are unicellular organisms. One of the amazing things about life on Earth is that across all of these organisms, the cells and parts of the cells are highly similar. For example, all cells have a lipid membrane that separates the outside from the inside. In eukaryotes, the cells also have organelles, such as the DNA-containing nucleus and the energy-producing mitochondria. When scientists look at cells in terms of their parts, they find that the molecules and organelles in yeast often act in similar ways to those in human cells. Researchers have used yeast to make great advances in our knowledge of how cells proliferate and divide. The cell cycle in yeast and other eukaryotes are highly similar, with stages including GAP1, synthesis, GAP2, and mitosis. During S phase, the synthesis phase, the replication of DNA in yeast is very similar to the process in humans. One of the topics studied by the Gao lab at ICOB is how DNA replicates during S phase. To study this, they blend traditional methods for studying yeast with modern technologies. Well-established traditional methods, like the use of mutants in complementation or synthetic lethality assays, have been extremely useful to identify the genes and gene products involved in synthesizing DNA. In addition, the Gao lab uses modern analyses, like super-resolution microscopy, to see how those molecules behave during S phase. In one project, the researchers used this very powerful microscopy technique to identify all of the spots where DNA replication is happening in a single yeast cell nucleus. They can see that within a single nucleus, many parts of the genome undergo replication simultaneously during S phase, and the timing of replication is highly uniform from cell to cell. In their ongoing studies, they're especially interested to learn how this process is affected when they mutate key genes that were identified from traditional methods. Studies like this can provide new information about a cellular process that's common to all eukaryotes. And in this way, scientists can learn big lessons from studying this tiny organism. If you're interested to learn more about yeast or the Gao lab, please check out the links in the description.